So our next speaker, Katie Bentley, is a lecturer, senior lecturer at the main institutes at the King's College and has her lab at Creek. Her first lab was at Harvard, and she had 2 satellite labs, one in Boston and one in Uppsala, hence my confusion as to what to mention first. And today she will be talking about cell dances, cell choreographies, how cells, either as individuals or in groups, sense their environments, make decisions, and eventually adapt. So the title of her talk is Active Perception in Collective Cell Patterning, and officially the stage is all yours.
Thank you very much. It's just been fantastic to be here. I've already had such a good time. I don't know if you can... Okay, here we go. Thanks. Let's see, does that work? Oh, yeah, okay. What an interesting bunch of people to be talking to. And it's true, you can't see anything once you're up here. There's a bright light in your eyes. So, yes, I'm going to talk to you a bit about an active theory that we're literally working on right now. So it'd be really nice to get feedback and hear from different perspectives on this idea we're working on of active perception in cellular collectives. So we're going to go down to cell scale now. So I actually started my career here at Sussex. I did my bachelor's and master's here in maths and computer science, doing the easy MSc course. Learning about evolution and adaptive systems. So this is why you're going to see quite a lot of computer simulations, which are the sort of mainstay of what we do in the lab. But I got kind of wooed into loving blood vessels unexpectedly, and ended up learning quite hardcore biology, and now have a wet lab as well. So we sort of meld this, sort of try and cross the disciplines, I suppose, of computer simulations and wet lab vascular biology. And the way that I got sort of sucked into this area was by being shown beautiful images like this down the microscope of blood vessel networks. And this is actually a mouse retina, which is used as one of the standard experimental assays in vascular biology, because these blood vessels grow out in a really stereotypical way from the optic nerve. This is a developing eye of the mouse. And you can actually see the blood vessels, they sprout. So this is a little, it might be easier with the, oh yeah, I'll deal with that. So that is one little blood vessel sprout that's just starting to come out. And this is called the sprouting front. And so what happens is they start out from the optic nerve, radiating out, growing this vascular network. And I'm going to show you a little bit about how this process works, because it just seemed incredible to me that endothelial cells that line your blood vessel wall are able to collectively coordinate in order to grow these well-adaptive structures. And every single one is different. It's well adapted to the local needs of that tissue as that developing eye tissue is growing. And what really struck me as interesting is that in disease conditions, and your blood vessels pervade almost every organ of the body, so they actually affect or are a part of a lot of different diseases. But in particular, this is retinopathy that we study, and the mouse model of retinopathy of prematurity that happens with newborn children that are premature. And what happens is the disease conditions seem to cause the endothelial cells to construct very different morphologies. So instead of that nice sort of familiar tree-like structure of a densely branched network, you end up with several, a lot of blood vessels that are actually very enlarged and bulbous, and then other regions of the eye that have absolutely no blood vessels at all, and it's actually very poorly branched. And trying to understand this switch in collective behaviour and why they would construct this different structure, that's then is very important for understanding therapeutics. And it's these abnormal vascular structures that emerge during this disease that actually cause mechanical traction, and they pull the retina off. So they are actually the key problem in causing blindness in this retinopathy. So what we want to try and do is understand how do blood vessels actually grow normally, what do the cells do normally, in order to try to sort of trick these cells in the disease case to start behaving normally despite the fact they're in a disease environment. But in order to do that, we have to actually understand how blood vessels grow normally, and it turns out there's a lot of open questions and people still don't really understand it. So this is the first few sort of key steps of the process, and then I'm going to home in on one of them and tell you about our theories of how this works. So angiogenesis is the name for the growth of blood vessels, particularly sprouting ones from pre-existing ones. So it's a particular type of blood vessel growth, angiogenesis. And it happens during development in your organs. It also happens if you cut yourself, you'll start doing angiogenesis. So your blood vessels will have to start growing and reforming that network. And what happens is if a tissue becomes low in oxygen, because of a wound or because it's just growing during development, it will start releasing certain signals. So the tissue starts to release pro-angiogenic and anti-angiogenic signals. It actually needs a balance of these different things. It's not just one signal. But we're going to hone in on one just to sort of explain it. But just keep in the back of your mind, there's actually quite a lot of complexity and multiple signals going on that these cells need to sense. And here's our pre-existing vessel lined with endothelial cells. And what happens is they detect this signal, and then a few of them, not all of them, just some of them start to respond. And they respond by changing shape. And quite dramatically they change shape and grow fingers. And they grow these, they're called Filopodia and dactylpodia, dactyl meaning finger. And so they were these sort of literally kind of ellipsoid cells around the vessel. And now some of them start to grow these fingers and protrude out, perpendicular to the vessel. And this is zooming in on a real one in the mouse retina. And you can see these fingers here. And they actually use them to, they're involved in this migratory crawling response that the cells will need in order to migrate and sprout away. And so these cells that do this are called the tip cell, or leader cells. So these are the highly migratory cells that will lead the following cells to form a new blood vessel sprout. Those sprouts actually quite quickly find each other through a process called anastomosis, which people don't really understand that well, honestly. But these finger shapes are very important, and they sort of find each other and form a loop. They fuse. And then they can support blood flow. And once you have that blood flow, you've oxygenated that little bit of tissue, so it's done its job. And what happens is, to grow a network, you just iteratively follow these rules. So you select some new leaders from these new blood vessels you've just created, and then you can start to build up a dense network. And so this sprouting front that first emerges in your tissues when they grow is actually a really regular meshwork. It's not got that hierarchical, sort of familiar hierarchical tree-like structure that we tend to think of with blood vessels. That structure occurs later, and so actually the mouse retina is a lovely model for this, because you can, in one snapshot image, you see the new developing bit, but also the older bit, because it's coming from the optic nerve. This developed a little bit earlier, and it's getting that tree-like structure with the wider vessels. This is the artery here, and the vein here, just a bit wider than the others. And that widening occurs in a process similar to the way rivers literally sort of, it's blood flow. So blood flow that's stronger down one sort of track just happens, will start to widen that region. If there's regions that are lower flow, they'll prune away. So it's really just like the way that rivers start to sort of reinforce certain regions and less so in others. And that's how you actually get the morphology of the tree-like structure. So we actually study this first step, just the sprouting, the initial plexus formation it's called, so just this regular meshwork. And we just focus in on this patterning process here. Why don't they all respond the same way? And it was a really interesting question. When I first started as a postdoc, that was the big question. Why don't they all just respond? But actually, it makes sense that only a few of them respond so that they can be the leaders and the other ones can be their followers. But the mechanism by which this works was then discovered, and it's called lateral inhibition. And it's actually a pattern formation process that occurs in many different tissues and different scenarios. But we found that, well, just before I started, really, my then supervisor was part of the discovery that this is what's selecting this pattern of tip cells. And I'm going to attempt to explain lateral inhibition to you, which is quite a complicated process. So we'll give it a go. Basically, we've zoomed in on two endothelial cells. Endothelial cells are always stuck together because even when they're growing and growing these new blood vessels, they need to keep adhered, stuck together so that the blood doesn't leak out, basically. So these are two endothelial cells that are adhered here on what's called a junction between two cells. This membrane here, this part of the cell, is exposed to the environment. So the environment becomes hypoxic. Maybe you've cut yourself, or maybe it's this bit of the eye tissue that's growing, and it starts to generate this protein signal, a growth factor. So there's an environmental signal, and the cells start to detect it. They detect it because they've got receptors on their cell surface. This is called vascular endothelial growth factor, or VEGF. It's actually the primary growth factor for blood vessel growth. So the receptor is called the VEGF receptor, but we'll just call it a sensor to be simple. And it actually triggers, so when this receptor chemically binds this growth factor, it triggers a chemical reaction and actually a lot of chemistry going on in signaling networks inside the cell. But being originally a computer modeler, I forget about all that. And I just draw an arrow. So this is good. And it then causes loads of complicated changes inside, gene expression changes inside the nucleus. And we'll just put the word genes. And we're only going to worry about one of those changes right now, which is that it upregulates a ligand or a protein called delta-like 4, we'll just call it D. Delta-like 4 becomes a signal that this cell can send to this cell, because this cell has a receptor that specifically binds that signal. This is called the Notch receptor, and this is a big signaling pathway, again, in a lot of different cell systems in the body. So it was interesting that blood vessels use this too. So this cell on the left tells this cell on the right, it sends it a message, and it's literally called a sender-receiver system. And it tells it something. It tells it, I've got lots of this D stuff. And when that happens, this triggers another chemical signaling cascade that changes the gene expression inside this cell. And what the key result of that is that this cell starts to downregulate the expression of this sensor. So basically, this cell gets activated by the environment signal and tells its neighbor, I want you to not have any sensors for this anymore. I want to see the signal. I don't want you to be able to see this signal. And what happens is, because they're both in this same environment, they actually both start doing this and they battle. They battle back and forth for who's going to be able to see this signal. And over time, one of them wins the battle and will have more of the receptor, the sensor. And it's interesting because that one that has more of this receptor is then able to make these shape changes, because that receptor is known to trigger the shape changes to create those fingers. And so this one will become the tip cell, the leading tip cell. It's got more ability to sense, it's inhibiting its neighbors, they'll become the followers. And it actually is now able to change shape and migrate and do the behavior. So that is the lateral inhibition process. Lateral inhibition, literally you just sideways inhibit your neighbors. And that's what generates this on-off pattern. So everyone in the field of vascular biology was very, very excited at this discovery and thought, this is brilliant, we've solved it. But what I kind of thought when I looked at this was, but nobody's talking about how long this takes. And blood vessel growth is actually a very, very dynamic process. But the field was using these static images. You know, there's no dynamics to them at the time. And hadn't really thought about how long this process might take. So this, what you're watching now, is blood vessels growing in a dish, in vitro as we call it, so just in a petri dish. You can put endothelial cells in. We've colored labeled them fluorescently with green and red, so you can see individual cells. And actually it was by virtue of doing this, we did this for a certain reason, but we hadn't realized that by virtue of color labeling them red and green, we would actually see that, you have to kind of pick one to watch, but there's one here if it starts again. The tip cell gets overtaken. So this leader tip cell isn't always at the tip. These aren't fixed phenotypes. We discovered they're actually phenotypes in flux. They can change their mind. So the one that's inhibited could actually take over if the conditions change. So it's a very dynamic, it's a time-limited process. So you have to oxygenate that tissue, otherwise, it's going to die. So they're going to have to solve this patterning problem very quickly. And they're going to have to be able to repattern if things change. And it's great that this word Umwelt come up, already, because the cell's Umwelt basically is going to change a lot as they move around in space during this process, which we think then changes who their neighbors are, so who's inhibiting them, who they're inhibiting. So they're continually battling and competing as they extend these sprouts. And it struck me that, if you've ever been in a committee, then you'll know it takes us intelligent human beings a very long time to come to collective decisions. So why are we assuming that this process would occur quickly? It could actually take quite a long time. And how are these cells solving a collective decision patterning problem like this on the fly all the time, really quickly? This is just from 12 Angry Men. It's a jury. They've got to make a binary decision, guilty or innocent. And it takes the entire film for them to make that decision. So how long does it take these cells to make collective decisions? So in order to start answering this question, because of my background, I guess I instantly want to learn by building. Just let's build a replica system, a computer simulation where we can really study dynamics and start trying to understand what's going on. And my background in adaptive systems led me to instantly want to use embodied agent-based modeling. So basically, I think we've touched on the idea of embodiment so many times, it's brilliant. But for anyone who doesn't know, embodiment is the concept that you really explicitly consider the body, and you consider your morphology, the shape, the structure, the localization of where your receptors are on that structure, because it constrains and obviously modifies behavior. And on a robot, of course, if you put your sensors at the front, or if you mess them around and put them in all different places, it's going to change how well it's going to move its wheels and get to different light sources. I think it's a really clear mapping to cells that you would use this approach because they have such a plasticity of form. They can change their shape really dynamically, and I'm going to show you some movies of that soon. And that, of course, then changes the position of their sensors, although they also have active abilities to change positions of sensors. They can literally traffic them around very quickly if they want to, put them over here or put them over there. They have this really, really incredible cytoskeleton, which is, unlike our skeleton, which is fixed, the cell skeleton can actively build over here at 1 minute and then deconstruct and build over here. So they can grow arms and legs wherever they want all the time in response to the environment. And they have a kind of mapping of a controller, you know, in a sense of robots. We think of this sort of neural network, perhaps thinking about the brain in general. The cell has this gene regulatory network inside the nucleus, which, you know, you could start to map across. So we think of cells with this same sort of common framework, and we described it all in this ridiculously named paper, if anyone's interested, to see how we map this approach to cell biology. And then we call it an agent because it's autonomously generating its own behavior. That's the only idea of what the word agent means. It's just any individual that can generate their own behavior. So this is the conceptual approach. And many years ago, I built this simulation software called the memAgent-Spring model. And we've used it a lot of times and extended it. We're building new software now, but this is the one I'll sort of stick with for the talk. The concept of this model is that a cell, just like your body is a society made up of lots and lots of cells, the cell itself is a society made up of lots and lots of smaller pieces, proteins, molecules, organelles. So, and being made up of smaller pieces means it's easier to move, actually, and change the shape. So in this model, each cell is made up of a collection of smaller individual agents, which we call membrane agents or memagents because they represent sections of the membrane. And then you can have a localized allocation of receptors and ligands and signals so that one side of the cell can respond to, so this little memagent might be able to respond to the environment, VEGF levels, the growth factor levels here, in a different way to the way the one does over here. So it's a completely decentralized model, because we believe that's actually how the cells work. They really do a lot of decentralized interaction with their environment, not needing to go into any kind of centralized processing. We later put springs in between these little memagents so that we could model the tension in this cytoskeleton. And that's why this looks a little bit more fancy, because it's got springs going on, pulling, and we can arrange little rules that then capture the way that these Filopodia fingers protrude out and extend and retract by creating new linked chains of these little agents and springs when the sensors locally activate. So that's when the sensors activate, they go pink, and then that triggers this localized response, Filopodia extending out. And the cell itself has some overall thing, actually has no concept of this, you see what I mean. But there is an overall summing up of all the protein levels, an activation that changes gene expression on a slower timescale. So what we can do with this model is then start to simulate vessels under different conditions and play around and start to try and understand what's controlling the dynamics of the pattern-forming process. So this first step, so here's a vessel. Each cell is made up of about 1,000 of these little memagents, and they're interacting with a gradient of this growth factor above them. And you can see that there's two cells here still battling a little bit, but they are basically able to get this on-off pattern. They started out all the same, but now they're creating that nice pattern where some of them activated their VEGF receptors more, they upregulate this delta signal, and they use it to inhibit their neighbors, which then pull in and retract those protrusions. In the simulation, we can have them actually sprouting and moving forward and looping, but for this one, I just wanted to show you the pattern forming. Because what I'm going to show you is a simulation where we've then reduced a particular parameter. So we've actually done a lot of studies across a lot of different parameters in this model, and then found that they have relevant experimental phenotypes. But I'm just going to show you this one because it relates to perception. So what we've done here is just reduce the probability of one of those finger-like protrusions to come out upon sensor activation. So when one of the receptors activates, you get this protrusion coming out, and we just turn down the probability of that happening. So they are still doing it, but just less so. And what we noticed was quite unexpected, that the pattern of selecting those tip cells is slower to emerge in this concept. And here, if you actually just put the frames of the movies going underneath each other, so time's now going downwards, then they do get there, they get this pattern of on-off selection, but it took them a lot longer. And this was really, really surprising to us because the dogma in the vascular biology field is that this notch lateral inhibition process selects tip cells, and then the tip cells have these fingers, and that's how they move. No one's ever talked about those finger-like protrusions being part of the earlier decision-making process. They're always the end result. And that really struck us as surprising when we had to really go back and think about, does this model just have a bug in it? Is there something wrong with it? Why would you mess up the end result and find that it changed the entire process? But in order to understand this, I think it's good to think about what are these Filopodia. And so these little protrusions that stick out, they're, as I say, locally activated receptors, and they cause actin, this cytoskeletal polymer, to polymerize, which gets bundled together, and they polymerize out and push the membrane out. And so you get rapid extensions out, and then if that sensor goes off, they actually rapidly retract. And when I say rapid, this is actually a movie that we got last week in the lab by Irena, who's fantastic. And she's labeled fluorescently the actin. So this is, you're actually watching the actin polymers forming in response to this growth factor in an endothelial cell in the dish. And you can see how quickly they come out and then quickly they go back. So these are rapid responses. And it made us think about, well, wait a minute, so they must be happening first then. It makes sense that they would happen prior to this decision-making process. So we started to think, well, why would it be slower without these protrusions? Why would it be slower for cells to battle back and forth? So if you think about where the VEGF receptors are on the surface, if the cell can't change shape in any way and it's just stuck, it can't grow these Filopodia, then the input signal by these two cells will be very similar because they're microscopic, they're right next, they're just in the same gradient, and the receptors are all just on this surface. You'll get the same basic input going into this system. But gene expression is slow. It takes on the order of hours to happen, whereas these Filopodia are very, very rapid in order of seconds. So we simulated this with a maths model. If you have just the delta notch passing, battling going back and forth, and it takes actually multiple battles back and forth for these cells if they have basically the same input. It takes a long time for them to battle before they can amplify that small input change into a decision. Whereas if you have Filopodia, which is basically a positive feedback of sensors and motors, the Filopodia activate sensors, they move the sensors forward, which then will, if they're in the right directions, keep them moving forward and keep the sensors activating. And then you can have a very rapid difference where one cell's got a lot more protrusions than the other, because they're quite stochastic, but this one might happen to just go in the right direction and get those reinforced. Then you get one with a much bigger input than the neighbor cell. And then that bigger input is much quicker to be amplified. So basically, they help the cells make a quicker decision. So we wanted to check whether this would actually happen in vivo. We started to feel confident it made sense. And I convinced my collaborator, Shane Herbert, at Manchester University, who works with zebrafish, to test the idea. So this is blood vessels growing in the zebrafish. They grow out from the dorsal aorta, and you see them sprouting up, and you see the little finger protrusions as they go. They've been well studied. This system is very widely studied. But no one had ever looked before it starts whether there's any Filopodia. Are they there before during the decision-making rather than while they're migrating? And actually, we found there are Filopodia sticking out before the tip cells are selected. And we were able to selectively inhibit those Filopodia and see what happens to the timing of tip cells coming out. And what we found was that, so this is showing you frames of a movie going down in a control normal case. So the dorsal aorta, and quite quickly you'd normally get sprouts coming out. First cell comes out, second cell like that. When you inhibit those Filopodia with latrunculin B, then it takes a lot longer before that first cell comes out, and all the sprouts eventually get there, but they're much, much slower. So just as the simulation predicted, and I thought this was a wonderful image. So this is showing fluorescently the activated cell. So the one that has the most sensor activation is showing up fluorescently as yellow. And that's always the tip cell, and you always get one cell selected going up as the sprout starts. This is in a latrunculin B case, so basically we've got the Filopodia have been inhibited, and they're finding it harder to make that decision. And you can see here where there's a sprout that hasn't come out yet, a collection of about 5 of them, all activated, still battling. So to me, this seemed like that committee that's struggling to make a decision. So just to finish, I wanted to sort of step back and say why we think it's important to simulate in biology, because I think it helps you to think about your system in a different way. And we started to realize that the reason this hadn't been spotted before, that there might be a role for shape changes and movement, in this decision-making process, and that it's actually critically important, it allows them to make the decision in the time limit available, is because the way in biology all diagrams are written, so the dogma of how we think in biology, is in a certain order, that you have an input signal that goes to a sort of chemical signaling pathway, decision-making and gene expression changes, and then they always write the behavior at the end. You always put movement, division, death for the cell. The behavior comes right at the end. And I'm just going to show you an example of this, but this is, I think, because biology is still stuck in a bit of a signaling and gene-centric way of thinking about the world. It doesn't think about the body and doesn't think about movement very, very highly. This is an example from a paper. It's really standard that you get, you have your receptors at the top, signaling input, VEGF there, and then here's that signaling cascade, the complicated signaling network that they think is doing all the work, and that's why it's getting all of the space in this picture. And then the really boring bit from their point of view is the movement, which is right down here. So cell migration changes when the gene expression changes. So you can sort of see how someone's thinking about the process, the way they've drawn this diagram, that it's all about the signaling, and the body and the cell movement, the behavior is right at the bottom. But actually what we're saying is that we need to flip this diagram, really importantly flip it, and put it time-ordered properly, because the movement happens first, and the body response is primary. It happens way before the gene decisions are made, because they are slower. So when we write it the other way around, and we actually put it time-ordered, there's that movement, the Filopodia occur first, because they are fast. You can see how there's space for a behavior and body in sort of informing, changing the quality of what's sensed in order to better inform the decision-making. So we think this is where the cells use sensory motor feedback, active perception, in order to this positive feedback between movement and sensing, very, very rapidly in order to inform better decision-making. And that the way that we draw those diagrams normally just stops you being able to think like that, because the movement's at the end. So, I just wanted to say that we published this first proof of concept of this idea in the first special issue on basal cognition, which Mike Levin, who's going to speak in a minute, was one of the editors of. So I think this is a really exciting field that's starting to emerge. And we're hoping to sort of really prove this idea that cells actively perceive in order to make better decisions. And just say thank you very much for listening, and thank you to my lab. We need a new lab picture, because that's all clearly in lockdown. Yeah, thanks.